Hey there, welcome to 17.5. We are talking about mutations, which will hopefully be a little bit easier than translation or our 17.4 notes. So I think we have about 6 slides to go, so let's get this done. Early in the chapter, we discussed changes in the DNA that affect long segments of our DNA, and we call those large-scale mutations. Large-scale mutations are going to be examples like exon shuffling, which would pair up a different active site to a different part of another protein, and so they cause variation, which can be beneficial, which can be detrimental. But now we're going to consider point mutations, and then later on in this chapter we'll consider large-scale mutations again in the last slide. But point mutations are small-scale mutations. They're a single nucleotide pair change, and these once again can be advantageous or they can be detrimental. More likely, it's going to be detrimental. It might be silent, and very rarely it can be advantageous. But when they are passed on, it is called a hereditary disease or a genetic disorder if it is not advantageous. And in order for it to be passed on, it has to be a mutation in the gametes. The sperm and the eggs have to be the point where the mutation occurs. If it happens in your arm and you have skin cancer because you spent too much time in the sun, that is not going to be passed on to your offspring. An example of a point mutation that is passed on is sickle cell anemia. So this is a point mutation in the beta globin. So you can see here where there should have been a GLU, there is now a VAL or a valine. And so when your red blood cells are made correctly, they have this round shape. And when they're made incorrectly, or if you have sickle cell anemia, they have this sickle cell shape, which makes the cells less capable of moving through your blood vessels. They get clogged in your capillary veins, and they get clogged in the joints. They cause joint pain, and they can even cause heart problems throughout your life. It is detrimental, but we have found where it can be beneficial. But that's for a discussion on a later date. So when mutations occur in portions of the DNA that are not coded into proteins, so these are our introns that we talked about in 17.4, there is no effect on the phenotype. But small-scale mutations that occur within the coding portion of the gene can be broken up into two sections. These are single nucleotide pair substitutions and nucleotide pair insertions or deletions. These deletions can have large-scale effects. A substitution is the replacement of one nucleotide pair with another. And so if we look here, we can see that it was supposed to be a G, but we put an A instead, and so therefore a T was paired instead. And this created a change. However, this change is silent because it was supposed to initially code for glycine. So if it was CCG, it was going to code for glycine. However, it's now CCA, and that still codes for glycine. And so we call this a silent mutation. And this has no effect on the phenotype. If the substitution is expressed, so we have this here, we have TCG instead of CCG. And so this is going to code for a different amino acid. And so therefore we call that a missense. So the phenotype then is altered. It may be negligible, it may be small, or it may be very large. It could be a disease, or it could just be male pattern baldness. Who knows? But that is called a missense. If this happens again, if a substitution happens again, but it codes for a stop codon. So here we have an example. TTC codes for an amino acid, so TTC would code for an amino acid. However, ATC codes for a stop codon. This is going to stop the amino acid from growing, or the amino acid sequence, or the polypeptide from growing any longer. And this is, well, this is a problem because this is going to leave out huge portions of the protein. If it happens at the end, it may be not a huge problem. If it happens towards the middle or the beginning, it could be a huge problem. It could render the protein completely useless. But if it's towards the end, it might be a negligible change. So we just don't know. And so here we're talking about our large-scale changes again. Insertions or deletions are an addition or a lack of a nucleotide pair. So here we have an extra A. So it was supposed to be TAC, TTC, but we have TAC, an extra A, TTC. What that's going to cause is for the entire frame to move, the entire reading frame to move over one. And that's going to change everything. So in this case, it actually created a stop codon, which is going to stop the amino acid growth or the polypeptide growth. And that could be, once again, negligible, or it could be a huge problem. Here, if it's happening here, it added, so it's going to change the entire amino acid sequence. So if we took out an A, so this is a deletion, we're going to take out an A, and so then we have one missing. And so the amino acid sequence for the entire rest of the protein is going to change. We call this a frame shift. And once again, these can be very detrimental. Finally, what causes these changes are mutagens. Mutagens, when we hear mutagens, we think cancer, because changes in our DNA can eventually cause cancer. Once we interrupt how the cell cycle proceeds, we're looking at potential cancer. And it's only a matter of time. If you keep adding mutagens, if you keep making changes in your DNA, you're going to eventually change how the cell cycle operates. So it makes sense that when you hear mutagens that you think cancer. And so mutations occur. They can occur anywhere in the DNA, RNA, protein process. So they can happen during DNA replication, they can happen during transcription, and they can happen during translation. If they happen during during replication, they tend to be a bit more harmful because you've modified the template. You've modified every protein that's going to be translated from that gene from here on out. Whereas if it's an RNA or a protein, so if it's transcription or translation mutation, you're really just changing one protein, and that's probably going to have negligible effects. So usually these things are changed or found in proofreading during replication or during transcription. But if they're missed, it's called a spontaneous mutation. And so physical, chemical, and even infectious diseases or infectious invaders in our body can cause mutations. And so we call these mutagens. And so we're going to classify these in 3 different ways. One is a nucleotide analog. A nucleotide analog is chemicals that are similar to nucleotides but pair incorrectly. So it's something that is similar to a cytosine, and it's similar enough that it can take its place, but it's not cytosine. And therefore, the G isn't going to be able to base pair, or when it's read, it's not going to be read correctly. So that we call a nucleotide analog. Some chemicals insert themselves into the DNA and interrupt the shape of the double helix. Um, an example of this would be a thymine dimer. And so a thymine dimer is when you have two thymines together, and when you have X-rays specifically, they're going to cause a double bond between these two thymines, and it actually interrupts the shape of the actual double helix, which can be very problematic. Finally, some mutagens can alter the bases that are present in the DNA strand. And when you change the bases, you change the base pairing properties. And so you actually change the way that the genome is read and transcribed. So that is it. We are done with Chapter 17. Have a great day.